So our next talk, we're getting into some of the technology-based solutions, I think, that we want to get into. And would you like to borrow the mic and be here, or do you want to use this microphone?
I'm going to use this mic.
Okay. So we have Professor Yoon coming from the Biomedical Engineering in the College of Engineering Department. And thank you for coming today.
All right, so is this on? How about now? Can you hear me?
Yeah.
My name is Jeong-Yeol Yoon. I'm from the Biomedical Engineering and as well as Biosystems Engineering. And I'm going to talk about the PFAS detection using the microfluidics and smartphone detection technology. So I've been working on the microfluidics, especially paper microfluidics and smartphone detection as handheld and portable solution for detecting many different pathogens and disease markers and chemicals and so on. This particular project is in collaboration with the Dr. Kelly Reynolds in the public health. And let's just talk about this. I don't think I have to explain further about what the PFAS is about, known as the forever chemical. And then it's actually a collection of chemicals. And if you're looking at the PFAS, then people usually start their research starting from PFOA, which is the simplest form. And when he actually submitted this paper, then we actually ended up with a really strict reviewer. And so he actually demanded, or maybe she, demanded to test for all possible PFAS forms, then the reviewer will be convinced. And then initially you only have the PFOA data. And so we added PFOS, and then the editor actually said that should be enough. Don't worry about the reviewer. The problem is this. When you're talking about E. coli, Salmonella, the Cryptosporidium, and norovirus, COVID, the malaria, Zika, you do have commercial antibodies available. Sometimes you do have enzymes available. Sometimes you have aptamers available. You do have nucleic acid sequence available. Do we have antibodies for the PFOA? Actually, the answer is no. And even if we have, people are talking about using molecularly implanted polymer as kind of a synthetic antibody to the PFOA. The problem is PFOA is not the only type available found in the environment. So we need a different approach. So I'm not going to talk about the problems of the PFAS, but the EPA Lifetime Health Advisory, and then when it first they wrote this manuscript, it was suggested 70 ppt. And by the time the paper was accepted, and then we found that they lowered it down to 4 ppt, parts per trillion. So the 4 ppt corresponds to 4 femtogram per microliter. We always use the term per microliter, not per milliliter, because we are only using few microliter of samples. So, concentration is sometimes confusing. If you are collecting like liters of water, then they always use the term per liter. And if you're using like a small tubes, like a 1 meter scale, then you usually use the term per milliliter. But for the microfluidics person like me, we always prefer to use per microliter, because you're only using 4 microliter of samples. But regardless, 4 femtogram detection is something that is almost impossible to be demonstrated on the portable platform, unless you enrich them or amplify them. So that's the one method, one challenge. And here is the available method available out there, HPC, MS, tandem MS. And that's probably de facto the most standard available method, but you need a lab. Some people are suggesting use pH indicator methylene blue. Obviously, they are not specific. It can detect anything. And people have talked about the molecularly imprinted polymer, but I don't think it's actually working because it lacks specificity. So we want lateral flow assay, like rapid antigen test. We don't have antibody. That's the another problem. And also, we need to demonstrate really, really low detection limit. So most commercial lateral flow assay has a detection limit down to maybe a few tens of nanogram per mil, which is actually still a lot higher than the EPA required minimum. So those are the typical lateral flow assay method, and which is just a single the paper strip. And we actually did a lot of work to improve the limit of detection down to a single virus copy level, which means for the norovirus, we're talking about the attogram scale. The trick is essentially in the lateral flow assay, you do have the target line where all the molecules are actually being captured, and you are collecting lumped signal from it. But instead, we're separating all the molecules throughout the channel and then observe them through smartphone fluorescence microscope. And then collect all the data, and then since you're counting, in this case, we are using sub-micron size fluorescent polystyrene beads or latex beads, and then actually count them. So we were able to demonstrate such a low limit of detection. This is also in collaboration with Dr. Reynolds, and we demonstrate the same with the COVID. The problem is, some people actually think that the method is too complicated. After all, it is a fluorescence microscope. Even though it is small, you have to place it, position it, and focus it. Okay? So we need a simpler method. So there are many challenges in detecting PFAS. So first, it has to be specific without using antibody. And it has to be simple, and it has to be real fast, and it has to be something that you do not need any extra equipment. So our approach is this. Rather than using just a single antibody, let's choose protein fragments or peptides as a collection. Okay? And of course, one protein may not be specific to one type of the PFAS, but if we actually approach them collectively and using it like a machine learning way, the pattern recognition, we may be able to demonstrate specificity. And then we're actually paying special attention to albumin because we just found an article published like a couple of years ago. Why PFAS causing cancer to human? Because the PFAS binds to albumin in your blood and basically perturbing changing the osmotic pressure in your blood and then letting the blood contents leaking up from your blood and then creating all sorts of problems, and then which is essentially leading to cancer. So we are interested in starting from the BSA as our starting material. And then, of course, when you are using BSA, then we can use a little bit similar molecules, like casein, very similar to BSA, but much, much smaller. And then we are also interested in lysine because they carry net positive charge, whereas the many PFAS carries a negative charge. So we're just going to collect lots of data, usually 3 different data, and doing the assay on the paper microfluidic chip, multi-channel, usually 4 channels, and then analyze them collectively. We may have to make a sensitive and specific detection. And what's happening here is this. So we're going to preload BSA, casein, or lysine to the paper, and then they're going to be captured within the paper pore because of their molecular interactions. When PFAS comes in, because the PFAS is a very strong surfactant, they actually snatch BSA from the paper substrate, and then they are actually moving towards the moving front, which is the air-water interface. Why? Because they are hydrophobic. And air is also hydrophobic substance. So when the molecules move towards the front, then surface tension will be lowered. Lower surface tension leads to the slower flow. That's actually proven by the classic Lucas-Washburn equation in capillary reaction. So what we need to do, we just monitor the flow rate by taking smartphone video. I don't need any microscope. I don't need any imaging analysis. I don't need to measure any intensity. And then it can be done under any environment. So this is just the stage monitoring the flow velocity or the flow distance over time. And we create this flow velocity profile, which is usually a square root function. And we collect lots and lots and lots of data. I'm not showing an entire data set. It's just one set of data, and there are like 4 different replicates of such thing. And then we found that we loaded the high concentration lysine, low concentration lysine, same thing with the casein and BSA, and then found that the BSA actually worked the best in making a distinction between DI water and PFOA concentrations. And this is the PFOS, which is a little less sensitive, but the results are pretty much the same. And then we tested many different concentrations of the PFOA, and then we can actually detect down to 0.01 femtogram per microliter, way below the EPA guideline. And this is the PFOA's results, and then the limit of detection is the same. We also spiked the PFOA into the effluent wastewater, which is a light processed wastewater, and then you can still detect 0.01 femtogram per microliter. So the real question is this: can it actually make a distinction from the other similar chemicals? And then, since PFAS are mostly surfactant, essentially, so I'm trying to make a distinction from anionic, cationic, and the strong acid and weak acid. So we are choosing SDS, Twin 20, and CTAP. And the results are a little mixed, but if we are actually using the combination of the BSA and lysine data, then we can make a clear distinction between PFOA, PFOS with the other surfactants. So these are the decision metrics. So right now, this model is very simple. Our learning database has only like a few tens of the data sets. But right now, we just luckily got the pilot grant support from the One Health initiative. And then I thank for the advisory committee and then reviewers. And then our goal is to expand this model, test it for many other proteins, protein fragments, and peptides, and build a huge database and test for a large variety of the PFAS materials. So that is the goal. And actually, if we can come up with these such decision metrics, we can choose maybe 2 or 3 different proteins or the protein fragments, and then we may want to actually use them as a remediation material. Just paper strips, load 2 or 3 proteins on it, let it pass through, and then it's going to capture small amount of PFAS material specifically while keeping all the others. And we are also interested in, if it actually works for the PFAS, I'm pretty sure it will work with the nanoplastics. Nanoplastics, there's a huge array of different materials. There's no general antibodies available for nanoplastics, but I'm pretty sure we can find similar tactics can be applicable towards the nanoplastic detection and potentially remediation. So that should be it. So thank you very much for your attention, and I'll be happy to answer any questions you may have.
We have time, I think, for one question. I have about 30 for you later, but for now, does anyone in the audience have a question? Okay, great. It's super interesting, and I can see a lot of applications to many things that we're concerned about. Zoe, yeah.
Yeah, have you combined this? I worked in a lab where they made current, they used current, combined flow and current to sort of provide a measurement too, or?
Yeah, so in biosensor world, we either use optical detection or electrochemical detection. Electrochemical detection requires the voltage and resistance, conductance, current, and so on. They are super simple. That's the benefit. But the downside is the limit of detection is usually few orders of magnitude higher than optical detection. So certainly we can implement it as a supplementary, complementary material or method. But in terms of lowering the detection limit, I actually doubt that would work.
Well, I was wondering if it combined.
Yeah, but we can certainly actually combine two different methods together.
Okay, thank you very much.